And so the histamine increases the capillary permeability. It allows more stuff to, essentially more stuff to leave the capillaries. So it enables the white blood cells to easily leave the bloodstream to get out to be able to attack the pathogen. Okay, so let's look at one more type of white blood cell. So the last one I have here is a monocyte. Okay, that monocyte will turn into a macrophage. That is a C. Okay, so that monocyte will turn into a macrophage, okay, and they can also become part of the adaptive immune response. Remember, the adaptive immune response is more specific, and these can become antigen-presenting cells. And so when they become antigen-presenting cells, they can trigger the formation of antibodies. So monocytes can turn into macrophages as well as antigen-presenting cells. So monocytes can be have both an innate response as well as an adaptive specific response. Okay, so now that we've got all these different kind of cells, let's see what it takes to actually trigger the phagocyte. So let's see what it takes to get that activated and get that going. Okay, so the response is going to be very similar to how it was in invertebrates. We've got these toll receptors. Okay, so you've got your toll receptors again. Okay, these are the TLRs here. Okay, so we've got the toll receptors that are going to be triggered. Okay, and remember, PAMPs are going to trigger these. Like these would be examples here of PAMP molecules. Remember, ones that are specific, they act as identification signature molecules. Okay, and so these PAMP molecules, again, are going to trigger our toll response. Okay, and they're, remember, our pathogen-associated molecular patterns. Okay, and that's going to trigger the response of the cell, which is going to be both an inflammatory response as well as it forms the vesicle. Okay, and it brings that molecule inside and is going to digest it. Okay, phagocytes can actually eat themselves to death. That's what pus is, when you've got an infected area, and so there's a lot of phagocyte activity going on. They're eating a lot of pathogens, okay, and basically they eat so much that they explode, okay, and that's what pus is, okay, and again, it's another what we've got here in vertebrates is another example of cell communication happening. Okay, here's my signal, okay, binding to my receptor, triggers my signal transduction pathway, triggers my response of both inflammation and the digestion of the engulfed particle. Okay, so far we've been talking about cells that go and attack the actual pathogen or bacteria, the invading molecule. Okay, so let's talk for a second then about natural killer cells. Natural killer cells will attack your own cells. So what they're going to do is they're going to attack both infected, and if they can find them, they will attack cancerous cells. So they're basically sacrificing one for the good of the many. Okay, so these infected cells will have, infected cells and cancerous cells will both have changes, remember triangle is changes to their cell membrane or their plasma membrane. And so the natural killer cell recognizes those changes. And so that triggers the natural killer cell to secrete chemicals into the infected cell or punch holes in their membrane. So it will either inject, so this is my natural killer cell here, and so it will either, like I said, it will either punch holes in the cell membrane or it will, wow, I cannot write tonight. So it's either punching holes into the cell membrane, okay, or it is secreting chemicals into the infected cell. Either way, it will essentially cause lysis of the infected cell. Okay, if it punches holes in the membrane, it encourages water to go into the cell, and so the cell will swell and eventually lyse. Okay, so natural killer cells, like I said, they're attacking your own cells, but they're only attacking hurt or infected cells, cancerous cells. Hopefully, and the goal would be so that those cells then cannot infect or spread that response anywhere else. Okay, so let's look at our chemical responses. So we've looked at our physical barriers, we've looked at our cellular response. So let's look at our chemical response and wrap all this up. So as these phagocytes become activated, they're going to secrete what are called kinins or chemokines. Okay, so they'll secrete the kinins or the chemokines, and what those will do is that they will increase circulation. So I'm going to increase my circulation here. I'm going to increase my capillary permeability. Remember, I'm going to let more things be able to leave the capillaries, get out to that infected tissue. Okay, so I'm going to increase capillary permeability. They also trigger a response, what's called chemotaxis. Okay, and so taxis is to move. And so basically what chemotaxis is that these cells move in response to a chemical. Okay, so they're moving in response to a chemical. So it will attract leukocytes to the area that it's injured. So those are our white blood cells. So that's why I have an increase here. My chemotaxis is that way because the more of those chemokines or kinins that I have, the more it's going to attract the leukocytes. So the cell is going to move in response to that chemical signal there. Okay, they can also affect the nerve, and they affect the nerves by actually making you feel a little bit of tenderness, a little bit of pain, make you aware that something's going on here. Okay, so this is one of our chemical responses, the secretion of these kinins or chemokines that are going to increase our circulation, capillary permeability, cause that chemotaxis to make the leukocytes move to the area, and trigger the nerves to make the area a little tender. Okay, so now let's look at another chemical called complement. Okay, so complement is formed when also in response to these invading bacteria. And so when the complement is activated here, so this is the foreign cell here, and it has these markers on the outside that trigger the complement activation. And when the complement is activated, it forms what's called a MAC. Okay, that's a membrane attack complex. You can see the MACs here that have been formed. So it forms this membrane attack complex, or MAC. And that MAC is going to act very similar to a natural killer cell. It's going to punch a hole in the membrane of the foreign invader. And so when it punches a hole in the membrane of the foreign invader, it triggers water to go into the cell. And so when that cell fills with water, that cell can end up exploding. Okay, so complement, again, it's nonspecific. It's an innate form of immunity. Okay, but it acts, like I said, very similar to the natural killer cells in the sense that it kills the cell by punching a hole in it, but it attacks the foreign invader, whereas your natural killer cell is going to attack your infected cell. Okay, your complement proteins can also coat the bacteria, so let's take a look at that and see what. Okay, so in addition to forming the MAC, okay, that can punch the hole into the cell, the complement activation can coat the outside of these pathogens, these foreign invaders. Okay, it'll work with the antibodies to do that. And so what it does is it allows the phagocyte to recognize it. Okay, so my phagocytic cell here, okay, is going to be able to recognize the pathogen because it has coated the pathogen with this complement activation. And so what that is called is that's called opsonization. Okay, so again, the coating onto the bacterial cell, you can see the IgG that's been added there. Okay, so as the complement bacteria works with this antibody to coat this pathogen, okay, that allows the phagocyte then to be able to recognize now the pathogen and go ahead and engulf it and take care of it. And that is called opsonization. It's marking that pathogen for consumption. Our next chemical is what's called interferon. Okay, interferons interfere with the pathogen. Okay, so interferons are secreted by the infected cell. Okay, so so here comes my virus here, and so my virus is infecting the cell here, and this cell is going to secrete the interferon. That's what these little yellow molecules are. And so those little yellow molecules head to the neighboring cell. So this over here is a cell that is unaffected. The virus hasn't gotten to that cell yet. It hasn't infected it yet. So this interferon is going to travel to the neighboring cell, and it's going to prevent that cell from being infected by the virus. Okay, so the interferon can't knock out the infection in this cell here that the virus has already gotten into, but it can help hopefully prevent that virus from spreading. Okay, so let's look, as we're on these chemicals, let's look a little closer again at histamine. Remember, histamine is secreted by basophils, secreted by mast cells, and it's going to encourage that inflammatory response. So let's take a look at what that inflammatory response is. So first of all, when the pathogen has been introduced to the body, okay, that's going to secrete our chemical signals that are it's going to release those chemokines, those kinins that are going to attract the various leukocytes to the area. Okay, and so as those leukocytes head to the area, that's going to release, like I said, they'll release the histamine. So we're attracting those basophils, we're attracting those mast cells, and so that triggers the release of histamine. And so when I've got an increase here in my histamine, remember I've increased my capillary permeability. So you can see now I've got red blood cells that are able to go out there, platelets that are headed out into the tissue area, so I can start clotting. I can start the clotting process. I can start sealing it off. That's one of the points of the clot, is I can now seal that area off and try to keep that pathogen localized right there. Okay. It also allows the leukocytes to get out there. So now those phagocytes can get out of the capillaries, so they can get out into the infected tissue. So again, the histamine has increased the capillary permeability. They're attracted to those chemokines and those kinins. Okay? And then those phagocytes can help start cleaning up the damaged cells, the pathogens that have come in. Okay? And when they're all done, the macrophages will actually secrete substances that are going to decrease the inflammatory response. So they'll secrete substances that will suppress inflammation and promote the actual repair of the tissues. Okay, so when all these things are happening with inflammation, we start to see an increase in redness. We usually see an increase in temperature to the area. Usually it becomes swollen, okay, and a lot of times it will also become painful when we have inflammation happening. All of these are signs of inflammation, okay, and they're all due to this increase in blood flow that we have to the particular area from the histamine. I'm going to get a change in color because I'm going to increase the blood flow to the area, so it's going to look a little redder. That's also going to raise the temperature. Remember, blood carries heat, so it's going to raise the temperature of that particular area. It's going to make it swollen because there's more fluid in the space. Okay, and that's part of what can also make it painful. Okay, it can compress nerves that trigger then that pain response. Okay, and all of these things are important for inflammation. The increase in temperature can impact your body's ability to heal. Okay, it can help speed up some of those enzymes a little bit. It can also be a temperature that's not conducive for bacterial growth. Okay, the swelling and the pain let your body know that something has happened. Okay, they may also render it immobile so that you can't continue to move that blood around and circulate it and push that pathogen off into other areas of the body. Okay, so the inflammatory response in small amounts is very good. It's when our body has chronic inflammation that we begin to see problems with it. Okay, so the last one we're going to talk about then real quick is a fever. Because a lot of times a fever will go hand in hand with an inflammatory response. So what we would need to decide with our fever is actually whether it is good or bad. Okay, because a fever is not always the emergency that we make it out to be. Okay, so when we get a fever happens because those cytokines are going to they stimulate the brain to make prostaglandins. Okay, so my brain is making prostaglandins. And if y'all remember what a prostaglandin is, remember that's one of those paracrine signals. So a paracrine signal is something that's acting locally. So it stimulates the brain to make these prostaglandins, and those prostaglandins are going to go to the hypothalamus. Remember, the hypothalamus is just another part of the brain. So I'm acting locally here. Okay, and it's going to alter where our body temperature is set at. The hypothalamus is where we measure our body temperature. Okay, and it will change the set point of that body temperature. And when it does that, When it raises that, it's going to constrict the blood vessels in the skin.